Convolutional neural networks. CNNs are a significant advancement in artificial neural networks, drawing inspiration from the structure and function of biological brains. CNNs differ from traditional neural networks by connecting small regions of the input layer to neurons in the hidden layer, enabling efficient capture of spatial information. They are commonly used for image analysis tasks such as scene classification, object detection, segmentation, and image processing. To understand how CNNs work, we will explore three key concepts: local receptive fields, shared weights, and pooling. But first, let's take a look at the biological brain. Eyes receive light and convert it into electrical signals. The retina is a part of the brain that develops from the same tissue. Light rays hitting the retina send signals to both sides of the brain through optic nerves. Chiasma is where the optic nerves cross. Visual signals travel from the retina to the lateral geniculate nucleus, LGN, which relays visual information from the retina to the primary visual cortex and processes information related to shape, color, motion, and other visual functions. The visual cortex in the occipital lobe processes visual information and is divided into 5 areas, V1 to V5. V1, also known as the primary visual cortex, receives visual input from the LGN. Early research suggested that the nervous system detects specific features of stimuli rather than creating exact sensory maps like a camera. This understanding influenced the development of CNNs, mirroring the biological principles of information processing in the brain. In the 1930s, Hartline and his team found that light falling on a small circular area of a frog's retina could stimulate an isolated optic nerve fiber, which he called the receptive field. The term receptive field was first used by Sherrington to describe the specific area on a dog's skin that triggers the scratch reflex. In 1953, Barlow at the University of Cambridge and Kuffler at Harvard University independently discovered inhibitory surround in retinal ganglion cells of frogs and cats, respectively. This schematic diagram illustrates the receptive field of a ganglion cell, which consists of an excitatory center and an inhibitory surround. Signals from the central area trigger excitation, while signals from the surround induce inhibition of the ganglion cell. This image shows a 2D array of retinal sensory cells. When rotated 90 degrees, the pink circles represent ganglion cells, with one of them shown to be connected to the sensory cells in the receptive field. In contrast to traditional neural networks, there is only partial connectivity between sensory cells and ganglion cells. Please note that this diagram is highly simplified. Current understanding suggests that mammalian photoreceptors connect ganglion cells through retinal bipolar cells. Receptive fields come in different sizes and degrees of overlap. In the fovea, they are small for sharp vision, while in the periphery, they are larger for better motion detection, but lower resolution. This balance ensures detailed central vision and broad peripheral awareness. In the 1960s, Hubel and Wiesel discovered the presence of specialized cells in the cat visual system that respond to specific visual stimuli, such as lines and edges. Their work was pivotal for understanding how the brain processes visual information and has influenced research on perception, plasticity, and developmental neurobiology. Let's take a look at a simple example of how the hierarchical organization of receptive fields in the visual pathway allows for the detection of geometric features such as straight lines. This is the receptive field of a ganglion cell. The axon of the ganglion cell connects to a V1 neuron through the LGN. By combining overlapping receptive fields in a linear manner and converging their signals onto the same V1 neuron, this neuron can detect a bright line in a specific position and direction. If the central regions of the receptive fields are illuminated, we can have the greatest response from the V1 neuron compared to other orientations of light stimulation. In summary, neuroscientists have found that the visual system processes images in stages, beginning in the retina and moving through layers in the visual pathway. The initial layers detect simple patterns like edges and lines, while deeper layers analyze more complex shapes and objects. Neurons in the visual pathway have receptive fields that are responsive to specific areas of the visual field. In 1979, Japanese computer scientist Fukushima developed neocognitron, inspired by neuroscience. Trained with unsupervised learning, neocognitron extracts local features of input patterns at lower stages and integrates them into global features. It uses lateral inhibition, shared weights, local connections, and pooling. Neocognitron is a key advancement in artificial neural networks for image recognition and computer vision, influencing subsequent models. In 1989, Jan LeCun and colleagues developed the Lennet convolutional neural network using the backpropagation algorithm to recognize handwritten digits. The Lennet 5 architecture, introduced in 1998, included convolutional and pooling layers with learned filters. CNN adoption increased in the late 2000s due to large datasets and powerful GPUs. In 2012, Alex Krizhevsky's AlexNet, a deeper network with more layers and parameters, set a new performance standard on the ImageNet dataset. In the next video, we will learn how CNNs work. Thank you for watching.